the, uh, Thank you for the kind introduction. Good afternoon, everyone. I'm Kihyun, and I'm happy to share our research finding with you today. Mm, mm, yes. So I'm from A-Star. A-Star is Singapore's public sector R&D agency. A-Star is located in Biopolis and Fusionopolis. Research institute doing biomedical science are located in Biopolis. The other institute doing physical science are located in Fusionopolis. Among over 6,000 staff, around 36% have come from over 60 countries. So we are quite international community. There are 9 research institutes in biomedical sector. So my institute, Bioprocessing Technology Institute, is one of the research institute, and we are focused on the development of a therapeutic modality such as proteins, cells, and nucleic acid, and development of a large-scale manufacturing technology of the therapeutic modality also. Yes. For example, last year, BTI set up Asia's first automated mRNA manufacturing facility in Singapore. So as you can see here, we made the automated system for in vitro transcription mRNA synthesis using the robotics system. And this facility is designed to produce up to 15 million doses per year. Okay. Yeah. So mRNA vaccines such as the Spikevax and Comirnaty helped us overcome COVID-19 pandemic. These mRNA vaccines are based on lipid nanoparticle mimicking the structure of a virus. These lipid nanoparticles are carrying mRNA encoding virus spike proteins, and after transfection, mRNA is translated into the proteins to induce humoral and cellular immunity in our body. So now pandemic finished, so many people wonder what's next for mRNA vaccines. US government in July 2024, US government uh, initiated a 5 billion project called the Next Gen to prepare for the next pandemic. So they decided some major research area. So one of them is a pan-coronavirus vaccines. So as you know, SARS-CoV-2 virus continuously mutating into new um, variant that escape existing immunity quickly. So people want to develop a universal vaccine treating multiple SARS-CoV-2 variants at once. Also, people want to develop long-lasting monoclonal antibodies because mRNA vaccines cannot be applicable for all the people. For example, cancer patients undergoing chemotherapy have usually weakly uh, attenuated the immune system, so they cannot develop active antibodies efficiently. So in that case, people want to directly apply monoclonal antibody into the patient to build up the immunity. Another area is the development of mucosal coronavirus vaccines. So I want to explain why. For example, you may remember some people got mRNA vaccine 4 times, but still got COVID positive. And at the time, many people ask the question, I still get COVID after vaccination. Do I need to still mRNA vaccination? So this is still, we have an unmet need for mRNA vaccines. Why this is happening? We need to understand the mode of action of mRNA vaccines. Usually, mRNA vaccines are given by intramuscular injection, as you can see here. So after IM injection, mRNA vaccine transfect the somatic muscle cells and then tissue-resident antigen-presenting cells such as dendritic cells and macrophages. And these transfected APCs are migrated into lymph node and educate the T cell and induce the B cell development. So especially in the APC, mRNA vaccines transfect and then they do endocytosis first, and then after membrane fusion, mRNA is released to the cytosol. mRNA is recognized by ribosome to translate to antigen, and these antigen proteins are digested by proteasome and then assembled to MHC. And then finally, MHC induce CD4 positive T cell and CD8 positive T cell response. So at that time, why people choose intramuscular injection? Because intramuscular injection is the most commonly used route for vaccine delivery. Traditionally, this route produces greater immunogenicity, and it allows easier access to the draining lymph node. So usually you can see the muscle site, intramuscular injection site is quite close to the draining lymph node site. Also, it is known to produce less severe local reactions such as irritation, inflammation, and necrosis. But this intramuscular injection usually produces a weak mucosal immunity. So one recent study, they checked the blood and then bronchoalveolar lavage fluid in the patient, and then they found in the plasma, neutralizing antibodies are produced very well. So as you can see here in the upper panel, you can see three groups highlight in red. One is two times two doses of mRNA vaccinated, and then three doses of mRNA vaccination, and then J&J's adenovirus-based vaccination group. So you can see this vaccination group produce a similar level of neutralizing antibody compared to convalescent group. So it's quite working well. But however, in the lung fluid, neutralizing antibody level is much lower than the convalescent group. So means convalescent group are usually infected by a virus and they recovered. They are already protected against the virus infection. So this means mRNA vaccination cannot produce strong mucosal immunity at this moment. Why this is a problem? Because if you think about how SARS-CoV-2 trans infect our body, so usually SARS-CoV-2 infected person generates a aerosol, and then this aerosol infecting another person by airborne pathway. And then also they produce a respiratory droplets, and then these droplets can be uh, can infect other people by direct contact or indirect contact. But the common thing is this viral infection happens through mucosal surface, such as nose, mouth, lung, and eyes. So that's why mucosal immunity is important. So current intramuscular vaccination usually produce high levels of IgG in blood. So currently, they cannot block viral infection. So if we develop mucosal vaccine, and if this mucosal vaccine works well, they will produce IgA antibody in mucus together with the IgG in blood, so we can effectively block the SARS-CoV-2 infection. However, this mucosal delivery of vaccine is very challenging because of the mucin. acts as a major barrier. So in the airway epithelium, um, mucin is produced in this kind of two layers. One is a mucous layer having around 10 to 15 micron thickness, and the periciliary layer having around 7 micron thickness. So periciliary layer has tethered mucins like this, and then they are usually providing the lubrication for mucociliary clearance. So these layers are continuously sweeping the mucus to the one side and then remove the pathogens like a virus and bacteria outside the body. And then mucous layer contains the secreted mucins. They can form a very highly entangled gel network for capturing the pathogens. So if you take a look the structure, so secreted mucin molecules have a linear structure like this, and then cysteine-rich domain, N-terminal and C-terminal domains are there, and then in the middle you can find the glycosylated mucin domain in the blue color. So this glycosylated mucin domain has many sialic acid. This sialic acid acts as a receptor for bacteria and virus. So they try if high density of sialic acid helpful to capture the virus and bacteria. So usually what happens is N-terminal group has many cysteine, and then this cysteine has a SH group, and then SH group forms disulfide bond based linkage. You can see here. So they have a SS disulfide bond linkage. That's why they can form a network structure. And then sialic acid usually has carboxyl acid group, and that's why they have some negative charged polymer chains. Mm. So tethered mucin molecule is looks like this. They have a transmembrane domain, and then also the glycosylated mucin domains, and then they have a branched mucin brush like this. Mm. So especially for mRNA vaccines, it's really difficult to penetrate this mucin layer because mRNA vaccine lipid nanoparticles are easily captured by mucin and then swept removed by mucociliary clearance well. So mucin glycoprotein has can induce multiple mode of interaction. First, it can form a physical entrapment due to the gel network. Also, it has a highly negatively charged sialic acid, so they can form electrostatic interactions. Also, the blue color, the main chain, where this polypeptide, they have a hydrophobic interactions. So this means mRNA LNP need to avoid all these three interactions well to penetrate the mucous layer. Also, when people try to deliver mRNA lipid nanoparticle uh, to the mouth with the nasal route, they found the mRNA lipid nanoparticles are highly inflammatory. So actually, they induce high inflammation in the lung area, and then even it can induce mortality. So as you can see here, depending on the dose of mRNA LNP, um, around 80% of the mice quickly died in one day. So usually 10 microgram mRNA LNP dose is considered the human equivalent dose in mice. They found that this nanoparticle really toxic. Also, it induced serious body weight loss during the treatment. So they found the very this strong pulmonary inflammation is the major challenge of mRNA vaccines. So previously, people tried to use mucoinert polymer coating to enhance muco penetration. So one popular choice was polyethylene glycol. So this PEG polymer uh, has almost no interaction with the mucin. They can avoid the um, entrapment of nanoparticles in the mucin. So the attachment of a PEG, this process is called the pegylation, can help a nanoparticle make nanoparticle mucoinert. So other polymers like poly 2-alkyl to oxazolene and then polyvinyl alcohol are also popularly used. So the common property of these polymers, they are hydrophilic and also neutral charged. For example, people modify the biodegradable PLJ polymer nanoparticles with the PEG. So in the case, in the human uh, sputum, PLJ nanoparticles cannot move in the sputum very well, but pegylated nanoparticles are moving very well, and then they had a very long uh, distance travel. So, so try people think this PEG acts as a stealth coating. For example, in, like a stealth aircraft. So this aircraft cannot be detected by enemy because uh, they have a special property, right? So they people found the PEG coated surface usually have a protein resistance. So in our body, if any foreign materials are coated by proteins, and then it's detectable by our immune system. But PEG coated surface can avoid the protein coating. That's why cannot be detected. So people usually think there are three main effects. One is a cell explosion effect. For example, uh, initially PEG chain has some volume, but after protein coating, this volume is decreased. Actually, this also leads to reduction of entropy. So this is not preferred. So they want to maintain this exclusive volume. Second effect is high chain mobility. So if a PEG chain is sufficiently long, they have a high mobility and repair the protein coating. Also, PEG has a high capacity to retain water molecule because they can form hydrogen bonding with the water molecule. So in that case, this extensive hydration can avoid the hydrophobic interaction with the mucin. That's why it can help mucin penetration. However, this PG coating has a drawback. Usually, it requires a relatively high PG density. So it's summarized in this picture. When nanoparticle has a low PG density, mucin still can bind to the surface of nanoparticle, and then nanoparticle can be trapped in the mucous network. So only when nanoparticles are coated with a high density of PEG, mucin protein cannot adhere to the surface, and then nanoparticle has a high efficient diffusion in the mucin network. So in a study, people found at least 5% of PG content is needed to effectively shield nanoparticle from interacting with the mucin proteins. So you can see mucin protein coating drastically decreased and then saturated here. So at least 5% is needed. But this is not so good for mRNA transfection. It's working well for small drug delivery because small drugs like doxorubicin can penetrate the cell membrane easily. That's why the cellular uptake is not the matter. But mRNA delivery requires a cell uptake and efficient endosome escape. So, for example, people try to coat uh, GFP mRNA loaded LNP with a different density of PEG. You can see 1% PEG to 5% PEG. So with increasing the PG density, the GFP transfection rate drastically decreased. You can see very weak green fluorescence in the cell. Also, people tried the in vivo delivery of pegylated mRNA LNP. So as compared to 1.54% PG lipid coated LNP, 5% group has much lower um, firefly luciferase signal in the lung. So they found with increasing the pH lipid density, so cellular uptake and transfection efficiency of mRNA greatly decreased. So this means we cannot use the same approach for mRNA delivery. So in our group, we tried to develop a liquid core lipid nanoparticle. So, unlike mRNA LNP having a densely packed core, this nanoparticle has a soft core, making it deformable under external forces for better mucous penetration. So, our hypothesis is the creation of this soft core liquid help to um, penetrate the mucous network because of the high deformability. So to form a liquid core, we selected the triolane because triolane is a natural uh, lipid found in the olive oil. And also it is known to have a good anti-inflammatory effect. So you may remember traditional mRNA LMP kill the mice in when delivered by nasal route. So we hope to because of the strong inflammatory effect. So we hope to use triolane to reduce the inflammation and they want to make a safer nanoparticle. Also, this lipid has a melting point around 5 degrees Celsius. So this means this lipid exists as a liquid at body temperature. So that's why we chose triolane. So we mixed these six different lipid components. And then we use the emulsification and solvent evaporation technique to make a nanoparticle. And then after adding the mRNA, we can easily form a mRNA-coated lipid nanoparticle complex. So under cryo-TEM imaging, we can detect the liquid core formation in the nanoparticles. You can see here. Mm. So we decide to choose two different ionized lipid and cation lipid in this system. The first one is ALC315. This is a ionized lipid in the Comirnaty's formulation. Another one is DOTMA. DOTMA is a cationic lipid used in BNT111 for cancer vaccine in the BioNTech. So we decide to change the ratio of ALC315 and then DOTMA. For example, ILN1 has only ionizable lipid, but ILN2 has a 3:1 ratio of ALC and DOTMA. And then with the and then we change the ratio to make a 3 and 4. And then lastly, we also made a cationic version of LNN by using only the DOTMA. So helper lipid like a DOP and then beta cytosterol we kept the ratio constant. And then in this case, we use the PG repeat at 2 weight percentage. So why ionizable repeat are important? Because ionizable repeat has an endosome disruptive effect. So usually ionizable repeat having this has this structure, tertiary amine containing head group and then linker and then hydrocarbon chains. Usually they have a two hydrocarbon chains. That's why it's summarized. It's like this. So when this LMP are located in the endosome, and then let's assume they just enter the endosome and they have a neutral pH. So in this case, ionizable lipid doesn't have a charge, but endosome membrane phospholipid has negative charge, and they usually have a cylindrical geometry, and then they don't have an interaction. So two membranes are just stable. But when endosomes become acidified, uh, you can see ionizable lipid start to form a positive charge, and then they start to interact with the negative charged endosome membrane phospholipid to form a cone-shaped ion pair. So this cone-shaped ion pair is called to have a inverted cone geometry, and they tend to adopt a hexagonal face like this. So usually in this case, membrane fusion happens, and then mRNA uh, is released to the cytosol after membrane fusion. Mm. So we understand the lipid nanoparticle need to have a cationic charge to disrupt endosome and then escape from the endosome. But question is how quickly do the nanoparticle acquire cationic charges? For example, we know nanoparticles after endocytosis, nanoparticles usually go to early endosome. They have a mild acidic pH like 6 to 7, but a proton pump continuously bring the proton into the endosome and then acidify the endosome. So they become a late endosome and lysosome. So question is when they are escaping from the endosome, because if they escape too late, um, RNA will be degraded in the lysosome by enzyme and then acidity. So we hoped nanoparticle escape from the early endosome and then deliver mRNA to the cytosol. So to check this behavior, usually people use a special chemical called the TNS. So this TNS has a negatively charged sulfate group here. So interestingly, this compound is not fluorescent when free in solution. When this compound bind to cationic surface of LNP, they start to show strong fluorescence. So here is the example. When LNPs are immersed in pH 10 solution, they don't have a charge. So, but when we decrease the pH from 10 to 4, LNP start to have a cationic charge on the surface because of the ionizable lipid. So in that case, TNS dye start to bind to the surface of LNP and then start to show strong fluorescence. So usually the middle point is called the pKa, the acid dissociation constant. So this is the parameter that determines the surface ionization property of mRNA LNPs. So we investigated a pKa of iLNN. For example, in the case of iLNN2, we detected a pKa around 5.9. So you can see uh, other composition are summarized here. With increasing the DOTMA to ALC 5 ratio, we found the pKa gradually increased from 5.5 to around 6.4. So interestingly, we found that this is a roughly matched with the uh, cell cellular cytosol pH, also early endosome pH and late endosome pH. Mm. So especially in ILN 1 to 3 have a pKa value in the range of early endosome pH, So in that case, we expect these nanoparticles have a potential to escape from early endosomes for cytosol delivery of mRNA cargo. So we confirm the nanoparticle size was gradually decreased with the increasing DOTMA to ALC315 ratio. And then zeta potential, the surface charge also changed. So when ILLN, we found the ILLN2 mRNA complex has almost a neutral surface charge, as you can see here. So we consider this formulation may be an ideal for mucous penetration because near neutral charge surface can minimize the additive interaction with the mucin protein. So next, we try to screen the mRNA translation potency in the um, lung cancer cells, A509 cells. So we use the luciferase assay, and then we encapsulated the firefly luciferase mRNA, F-loop mRNA in the LNP. So if a transfection is successful, luciferase is expressed, and then when we added the luciferin, and then we can see a strong light from the cell lysate. So as you can see here, our nanoparticles uh, sh- among the tested formulations, ILLN2 mRNA complex showed the strongest luciferase activity. And interesting, this is higher than the benchmark LNP, ALC LNP. So this ALC LNP was prepared using the same lipid composition as that of a Comirnaty's mRNA vaccine. Also, all the formulations didn't have a cell toxicity. Mm. We also found uh, our nanoparticles can do endosomal escape. So to confirm the endosome escape, we use the Cy5 mRNA conjugate. Cy5 has a red fluorescence signal, and also we stain the endosome using lysotracker. Lysotracker has a green fluorescence signal. So if nanoparticles are still in the endosome, you can see the yellow color because of the co-localization of a red signal and a green signal. So in the case of AS-LNP, after 4-hour incubation, we still see a lot of yellow colored spots in the cell. So this means that these nanoparticles still entrapped in the endosome. But when we tested the iLN2 mRNA, they successfully escaped from the endosome. They try most cells has these red colored spots. And then quantitative analysis also showed our nanoparticles have a significantly higher endosome escape. So to check the mucous permeability, we set up the transfer mucous diffusion model. So this model uh, is prepared like this. In the transfer, we added the uh, mucin layer, and then we added the DII fluorescence dye labeled the mRNA LNP, and then we can check the fluorescence increase in the receptor chamber filled with the pH 6 cell line. So we use the pH 6 cell line because we wanted to mimic the acidic pH in the mucosal area. So as you can see here, ILLN2 mRNA complex showed much faster mucous penetration compared to cationic version of LLN and also ALC LNP, Comirnaty formulation. We also tested the effect of a liquid core by removing triolane from the ILLN2. So as you can see, ILLN2 lacking triolane has much slower mucus penetration compared to the original particle. So we think maybe the high deformability of ILLN2 contribute to the enhanced mucus penetration of ILLN2 mRNA complex. So we apply the efflux mRNA loaded LNP to the nasal area of the mice. So after 4 hours, we did the IBC imaging to check the luminescence from the luciferase expression. So as you can see here, ALC LNP was not so effective in delivering mRNA to the nasal cavity, but we detected a very, very strong signal from ILLN to mRNA complex. Cationic version has some moderate signal from the nasal cavity. So, and we also harvested the organs like a nose and lung, spleen, liver, kidney, and heart. And then we found that the signals are dominantly detected from the nose and lung. And then we found um, our formulation induced the 64 greater mRNA translation in the nasal cavity compared to ALC LNP. And then we found interestingly, we also found the cationic version of LNN tend to go to trachea area more. So you can see here is a trachea area as compared to ILLN2. Cationic version tend to accumulate in the trachea, although we don't know the mechanism. So we try to check intranasal vaccination effect. So we use the uh, PBX1010 mRNA. This mRNA was developed by a US-based company called Provexus. This mRNA is codon optimized mRNA encoding SARS-CoV-2 Delta variant spike protein. And then surprisingly, we found the broader protection in the mouse after 3 dose vaccination, and it also uh, shows broad protection against the Delta and Omicron variant. So we decide to give this mRNA at 10 microgram per mouse dose, and then prime dose was given at day 0, and then booster dose was given on day 21, 3 weeks interval. And then 1 week later, after 1 week after booster dose, we harvested the organ and then tried to uh, check the immunity. So, in collaboration with the A-Star ID Labs, we did the IgA and IgG measurement. So we found in the nasal wash, ILLN2 mRNA complexes induce the higher levels of anti-spike IgA and IgG compared to ALC LNP. ALC LNP didn't produce a significant level of antibody in the nasal wash. So we think maybe this nanoparticle penetrate the nasal mucosa and then reach the antigen-presenting cells. So maybe transfection of antigen-presenting cell produce the antigen, and then it helps to recognition by CD4 positive T cell, and then activation of IgA secreting B cell, and development of a plasma cell. This is a hypothesis. We also made some unexpected findings. For example, um, despite the detectable mRNA translation in the lung, both ASLNP and ILLN2 fail to generate anti-spike IgA in the bulb, in the lung fluid. Also, despite the lower mRNA translation in the lungs, ASLNP induced the higher IgA production in bulb compared to ILLN2 mRNA complex. Lastly, despite the very different mRNA translation profiles, ASLMP and ILN2 mRNA complex induce similar level of IgG production in the serum. So we still uh, don't know why. So this requires uh, we really wanted to know what was happening. So we tried to study the literature. So actually, according to literature, we found the mRNA translation does not well correlate with the mucosal IgA response. For example, in this paper, they developed the polymer mRNA complex nanoparticle and then deliver the mRNA to the lung using this vaccination um, timeline. They found this nanoparticle induce a strong signal, efflux signal in the lung, but surprisingly, no IgA was detected in the mucus. Only IgG was detected at that time. So we think that this may be related to NERT-mediated mucosal immunity. So NERT is the nasal associated lymphoid tissue. For example, in the mouse, NERT is located around this area. And then in the histology, we can see this high immune cell uh, dense structure um, called the NERT. So usually after the vaccination, antigen is uptaken by the dendritic cell in the NERT, and then they migrate to the cervical lymph node, and then bind to CD4 positive T cell, and then migrate to thoracic duct, and then the IgG secreting B cells usually coming to the effector site, in this case, lung area, and then secrete the dimeric IgA. Yeah. But actually, what we don't know is which cells are transfected. For example, in the case of a lung transfection, of course, we can see macroscopically lungs are transfected well, but we don't know which cells, which immune cells are transfected. This is one question. And then we also don't know the the migration of a IgA secreting B cell to the effector site is uh well uh well done or not. We also don't know. So they tried in the paper the authors suggest maybe additional signal may be required to effectively bring the IgA secreting B cell to the effector mucosal site. So they try, maybe this is a potential reason for the unexpected finding in our study also. Encouragingly, we found the ILLN2 had a better tolerability compared to ALC-LNP. So as mentioned before, ALC-LNP Comirnaty formulation killed the mice. For example, 50% of the mice died within 4 days after vaccination. But our formulation didn't kill the mice. Also, it avoided the body weight loss. So although some mice survived, they had some body weight loss, means they had some systemic toxicity. We collected the blood and then tried the clinical chemistry, and then we found the hyperproteinemia was caused. And then according to the literature, maybe this symptom is caused by inflammation. So we detected the total protein and then mainly albumin proteins are increased in the ASLMP treated group. So maybe this is related to the inflammation. On day 7, we collected the blood to check the pro-inflammatory cytokine, TNF alpha. We found the only ASLMP induced the high level of TNF alpha, means they induce some inflammation, systemic inflammation. And on the end point, day 28, we harvested the lung and then did the H&E histology. And then also we checked the Ly6G to check the number of neutrophils in the lung. So first we found some fibrosis occurred in the lung of mice treated with ALC LNP. And then the number of neutrophils in the lung also increased. So this is also another sign of inflammation. So um, it's not, although it's not conclusive, but we think the addition of a triolane, the anti-inflammatory lipid, may be helpful to reduce inflammation in the case of IRLN2 mRNA complex. So in conclusion, we developed the liquid core lipid nanoparticles capable of delivering mRNA cargo across nasal mucosa. And then we think our study may provide a useful insight into design of effective intranasal mRNA formulation for various biomedical application. So our dream is to develop effective intranasal mRNA formulation for easier delivery of mRNA vaccine. I'd like to thank Dr. Ian Yang, my uh, former supervisor in BTI, and then my colleagues, and then collaborators in ID Labs and NUS, and also grateful to IFPP and Prepare Grant funding. Thank you so much for your kind attention.
네, 박사님, 네, 재미난 강연 감사드립니다. 일단 제가 질문을 먼저 받을게요. 아마 저도 그렇고, 김우석 교수님도 질문이 왠지 있으실 것 같기는 한데, 지금 항상 저희 보면은 이렇게 재미난 강연을 해주시면은 너무 학생들 질문이 많아서 시간이 너무 지체되는 경우가 있어서 일단 학생들 질문을 먼저 받는 게 좋을 것 같습니다.
네네.
잠시만요. 제가 이 네, 먼저 이유라 학생 음소거 해제하시고 질문하시면 될 것 같습니다.
네, 교수님, 흥미로운 강연 감사드립니다.
네.
일반적으로 mRNA 백신을 근육에 주사하는데 특이하게 뮤신의 mRNA 백신을 나노파티클로 백신을 투여하는 걸로 이해를 하였습니다. 이렇게 근육이 아니라 뮤신에 투여를 하게 되었을 때 근육에 주사하는 거와 대비해서 특이하게 작용하는 장점이나 단점이 특별하게 있는지 궁금합니다.
Yes. So, she asked what is the potential advantage of a nasal delivery compared to intra muscular injection. So I think this is related to the this slide. For example, after intramuscular vaccination of injection of mRNA vaccine, people found IgG antibodies are generated in the blood. So they don't detect this IgA mucosal antibody in the nasal area. So in that case, we cannot block the viral infection. That's why people still get COVID after many times vaccination. So we wanted to deliver mRNA vaccine to the nasal area because we wanted to boost the mucosal immunity. So it's in the literature, it is well known. If we deliver uh, antigens to the mucosal site directly, we have a higher chance to boost the mucosal immunity in the deliver the mucosal site. That's why we wanted to deliver mRNA vaccine to the nasal mucosa. And then we found actually our result found uh, in the nasal wash, we detected the IgA, as you can see. Yeah, but actually we deliver mRNA vaccine to the nose. That's why we detected the IgA in the nasal wash, but we couldn't detect the IgA in the lung fluid. So this is still a weakness. So we, yeah, that's why we wanted to boost the mucosal immunity. This is the main reason why we chose nasal delivery. Thank you.
And Sanjalbek Habibullah, yes, your turn. Please turn on your microphone and you can ask.
First of all, thank you for your presentation. I have two questions. The first one is the all types of the ILLN2 they have the same mucosal immunity or the anti-inflammatory quality because they have the same quantity of the cytosol and the triolein. And the second question is, can we use ILL and 2 mRNA complex for the influenza vaccination? Because the children cannot the intramuscular injection, and this disease repeated in the winter many times.
Oh, okay. Okay. So about your first question, we made five different ILLN
Yeah.
formulation, as you can see here. They had the same amount of beta-citosterol and triolein. So in terms of anti-inflammatory effect, I guess they may have a similar anti-inflammatory effect. But we didn't test all the formulation to the mice. So it's still not conclusive. Yeah. Uh, so because of a limited resource, we had to choose the best performing nanoparticle for in vivo study. That's why we based on the mucosal, based on the in vitro screening, and then mucous permeability, we found the ILLN2 was the best candidate. That's why we chose only this compound and then cationic version for the in vivo study. Yeah. And then about your second question, I believe this nanoparticle can be used for um the influenza vaccine also. Uh, but we didn't test the influenza vaccine antigen loaded mRNA. And then also it's not conclusive because people know uh, mRNA uh, uh, it depends on mRNA length. So usually when people formulate lipid nanoparticles, yeah, we need to consider the length of mRNA. So people know if the mRNA is very, very long, it's difficult to encapsulate in the lipid nanoparticle, also difficult to deliver to the mouse in vivo. So I I think yeah, to confirm the real effect, we really need to do real experiment. Yeah.
Thank you.
Thank you. Thank
And
you.
sorry, last one. Yeah, the combination of the dotma and the three, through 3 and 1 in the second mRNA complex. Yeah, here. Uh, can we change the combination of the according to the pH level of the mucosal part of the people because we know the different place of the people they have the different pH. It depends on the region, climate, and in this case, can we change the this two combination?
Oh, yes, I think so. So actually, as you can see here, we found by changing the Rotuma to ALC315
Yeah.
ratio, we could modulate the pKa of a nanoparticle. So as you mentioned, in our body, depending on the location, the mucosal pH different. So maybe
Mhm.
nose pH around the 6, but maybe the other location mucosa
Mhm.
may have a different pH. So I think we can design this nanoparticle according to real application site. Thank you, thank you.
Thank you for answer.
아, 박사님 질문이 되게 많이 올라왔는데 괜찮으실까요? 시간이 가능하신가요?
예, 저는 괜찮습니다.
네,
네.
지금 손든 친구들까지만 질문을 하는 게 좋을 것 같아요. 왜냐면 지금도 굉장히 많은 친구들이 흔들기를 하고 있어서 자, 더 이상 이제 추가로는 손들기를 해주지 말아주시고, 자, 김설인 학생 하시면 될 것 같습니다.
교수님 강의 잘 들었습니다. 제가 비전공자라 이 분야를 정확히는 모르는데, 이 연구의 궁극적인 목표가 비강 백신 개발인지, 또 이런 점막 백신 기술이 코로나 외에 다른 호흡기 감염병에도 활용될 수 있는지 알고 싶습니다.
Ah, yes. So her question was um, whe- when whether this technology can be used for the um, airborne infectious disease. And then I think it's possible. And then our initial goal was to uh, develop um, develop effective nasal mRNA formulation because previously people wanted to deliver mRNA vaccine to the nasal route, but they found the mucosal IgA production was very, very low. So that's why we wanted to produce. Also, another issue was the safety. So a mouse killed by the nasal vaccine. That's why we wanted to use anti-inflammatory lipid to reduce the toxicity. And then I think this technology can be used for other airborne infectious disease. Uh, at, yeah, just the need because mRNA vaccine can be flexibly designed to produce a specific antigen against the different airborne infectious disease. So we can easily design the formulation. Mm. Thank you. Thank you.
감사합니다.
네, 다음 온지수 학생 마이크 켜고 하시면 될 것 같습니다.
들리시나요?
아, 네.
네, 흥미로운 강연 잘 들었습니다. 제가 엑소좀 쪽을 이제 하고 있어서 좀 더 흥미롭게 들었었는데요. 한 두 가지 정도 좀 크게 질문이 있습니다. 먼저 제가 LNP를 알기로 마이크로 플루이딕스로 두 상을 이제 혼합을 해서 만드는 걸로 알고 있는데, 보통 그렇게 하는 걸로 알고 있는데, 지금 나살로 쓰는 이 LNP 같은 경우에는 mRNA를 LNP를 만들고 나서 이제 나중에 포스트 로딩 하는 식으로 하신 것 같아서 왜 이런 식으로 했는지 궁금하고요. 그리고 이렇게 하면은 이제 로딩 효율이 좀 떨어질 것 같다는 생각이 들어서 로딩 효율을 혹시 보셨는지가 궁금합니다. 그리고 한 가지 더 있는데 그 제가 양이온성 리피드 캐티오닉 리피드 같은 거를 사용하게 되면은 어 이게 이제 리포펙타민처럼 세포 독성이 있어서 그 잘 안 쓰는 걸 많이 쓰지 않는 걸로 알고 있는데 어 지금 마지막에 독성 결과를 보시면은 어 기존에 있던 벤치마크보다 좀 더 지금 개발하신 LNP가 좀 더 독성이 낮게 나타났는데 혹시 왜 독성이 덜 나타났다고 생각하시는지도 궁금합니다.
아,
어,
yes,
어,
yes.
그,
So,
네,
네.
혹시, 그리고 제가 좀 빨리 피드백을 얻고 싶어서 한국말로 답변이 되시면, 네, 그렇게 해주시면
아,
감사하겠습니다.
네네. 어,
감사합니다.
어, 그래서 yes. 아, 한국말 답변해도 되나요? 그럼 그,
네,
아,
감사합니다.
예. 그래서 제가 테스트를 할 때, 인비트로 스크린을 할 때 썼던 ALC LNP 같은 경우에는, 이제, 코머디티 백신하고 똑같은 포뮬레이션으로 마이크로 플루이딕스로 만들었어요. 근데 이제, ILLN2 같은 경우에는 제가 이제, 리퀴드 코어를 만들어야 돼서, 이제 부득이하게 다른 방법을 쓰게 됐고, 왜냐하면 이제, 이 리퀴드 코어를 만들기 위해서 제가 이제, 중간에 어, 이베포레이션 과정이 있는데 마이크로플루이딕스 머신에서는 이런 이베포레이션 과정을 구현하기가 조금 힘들어서 제가 다른 과정을 쓰게 됐습니다. 어, 그리고 어, 두 번째 질문이 뭐였었죠? 두 번째 질문이
두 번째는 캐티오닉 리피드를 사용했는데도 독성이 좀
아,
더 덜 나타난
예,
이유가
예.
궁금해요.
이제 그 케타이온 리피드가 아요나솔 리피드보다 독성이 더 강한 건 맞는데 그렇다고 해서 이제 클리니컬에서 이제 전혀 못 쓴다 그건 아닌 게 이제 여기서 보시면 보트마 같은 경우에는 BNT111 포뮬레이션은 이미 사용이 되고 있거든요. 그리고 이제 휴먼 클리니컬 트라이얼 중이고 그 파이저 백신을 같이 개발했던 바이오엔텍이라는 회사에서 이 BNT111에 도트마를 써서 휴먼 클리니컬 트라이얼 중인데 독성은 이제 괜찮다고 나오고 있습니다. 그래서 이제 그 독성을 결정하는 거는 단순히 그, 그 양이온성 지질이 얼마나 많이 들어갔냐, 보다는 그 나노파티클이 이 몸 안에서 어떻게 대사가 되고 어떻게 방출이 되고 그런 걸 다 감안을 해야 될 것 같아요. 그래서 심플하게 그 양이 많다 적다로 이제 그 계산이 되지 않고 그 제 생각에는 이제 제가 했던 동물 실험에서 왜 이제 독성이 적게 나왔는지는 그 제가 생각할 때는 이제 ALC LNP에는 트라이올렌이 없어요. 그런데 ILLN2에는 저희가 트라이올렌을 이제 소량을 넣어줬잖아요. 근데 트라이올렌이 그 엔티 인플라메토리 효과가 있어서 이제 이 ALC LNP가 일으키는 그 인플라메이션에 의한 독성을 줄여준다고 이제 어, 생각은 하고 있는데 그것도 이제 정확하게 저희가 이제 디테일하게 이제 분석을 하진 않았습니다. 네,
좋은 답변
감사합니다.
감사합니다.
네.
네, 김요한 학생 질문하시면 될 것 같습니다.
네, 교수님, 박사님 들리시나요?
아, 네.
네, 그 두 가지 질문이 있는데요. 일단 그 인트로에서 소개해 주셨던 것처럼 이게 최근 백신 개발이 휴머럴 이뮨 리스폰스랑 셀룰러 둘 다 이제 가져가는 방향성으로 또 발전을 하고 있다고 생각을 하고 저도 이제 그런 것들을 연구를 하고 있는데 그 지금 발표에서 말씀해 주신 것처럼 휴머럴 이뮨 리스폰스 같은 경우에는 이렇게 확인하기 위한 툴들이 많이 사용이 됐다는 것을 알 수 있었습니다. 근데 특히 마우스 모델을 사용하신 겸 해서 이 더 많은 샘플링들을 하실 수 있었을 것 같고, 그다음에 에펙터 사이트라고 말씀해 주셨던 렁이나 그리고 뭐 날투와 같은 이민 오organ에서 어 안티젠 스페시픽한 이민 셀들을 이제 팩스 같은 툴로 확인하실 계획이 있는지가 첫 번째로 궁금하고요. 그 다음에는 이 LNP 같은 기존의 이제 mRNA 백신은 어주번트가 같이 사용되는 것들로 알고 있고, 이런 어주번트들이 보통 프로인플라마토리하게 반응을 유발한다라고 공부를 하고 있었는데, 지금 소개해주신 LNP에 사용하시는 트라이올린 같은 경우에는 안티인플라마토리한 성격을 갖고 있다고 하셔가지고 이런 것들이 백신 에피커시에 나중에 조금 영향을 끼치지 않을까 하는 생각이 들었습니다. 이 두 가지 답변해 주시면 감사하겠습니다.
아, 네. 그 사실은 그 mRNA 죄송합니다. actually 어 네, he asked the question about the adjuvant effect. So I want to show you this slide. Yes. Yeah. So usually the protein vaccines require adjuvant. But in the case of mRNA vaccines, mRNA itself can activate the toll-like receptor. For example, toll-like TLR7, 8, or 3, and then it activates the type 1 interferon pathway to produce cytokine, beneficial for the boosting this humoral immunity. So usually mRNA vaccine doesn't require adjuvant because of this reason. Yeah. 첫 번째 질문이 다시 한 번만 말씀해 주시겠어요?
그
죄송해요. 예.
마우스 모델에서 팩스 같은 이제 셀룰러
예,
이미지
예. And then his first question is about the um, facts or another analysis of immune cells in the mouse model. Um, Actually, I discussed this one with the ID Labs collaborator. They are doing infectious disease. They are more expert than me. But actually, they said it's quite difficult to harvest immune cells from the nasal cavity of a mouse. First, the volume is very, very small, and then it's possible to collect nasal wash, but it's maybe we can collect only the surface located immune cells rather than the immune cells located behind the epithelium. So usually you can see the structure. Oh yeah, yeah. You can see, for example, here is a nasal epithelium, right? So maybe we can, if we collect nasal wash, we will collect the mucous together with the immune cell. But these immune cells are not representative of a whole immune cell. So we need to collect the immune cells behind the epithelium. But in the nasal cavity, it's very, very challenging. So according to my collaborator, that's why we couldn't do the detailed analysis. Mm. Thank you, thank you.
나-
네, 다음 강은혜 학생 하면 될 것
네, 안녕하세요. 교수님 강연 너무 재밌게 잘 들었습니다. 제가 이제, 궁금한 부분은 두 가지인데요. 이제 앞서 질문들이랑 좀 비슷할 수 있는데, 어, 이제, 최근에는 이제 편의성을 위해서 먹는 약으로 이제 많이 개발되는 걸로 알고 있는데, 이제 제가 단순하게 생각했을 때, 이 비강으로 백신을 그 마우스에다 할 때 어려울 것 같아서요. 그래서 이게 어떻게 정량으로 들어가는지 흘릴 수도 있고 확인할 수 있는 방법이 무엇인지 그리고 어떻게 또 코로 맞을 백신을 어, 개발할 생각을 하신 건지 궁금하고요. 두 번째는 어, 그, 이 슬라이드에서 이제 코로나 바이러스로 테스트 하신 걸로 봤는데 이 ILNN, 이 ILLN 이 기술이 그 인플루엔자나 RSV 같은 그 다른 호흡기 바이러스에서도 혹시 테스트 해보신 적이 있으신지 궁금합니다.
Ah, yes, yes. So her first question is about the 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 comparison between the nasal route and the oral route. So actually, in the case of mRNA, it's very, very challenging to deliver mRNA by oral route because the usually stomach has a very, very strong acid and then enzymes, maybe the even nuclease. That's why I guess mRNA will be digested in the GI tract. So they try many people don't try oral delivery of mRNA. Uh, in the field, people choose the nasal route um, as compared to oral route more. Uh, and then in the actual mouse study, we have to give anesthesia drug to the mice to make them sleep. The mouse need to sleep and then don't move. And then after usually we give a 10 microliters volume of a mRNA vaccine, little bit, and then we pinch the paw. And then after pinching the paw, mouse gets stimulated to breathe. And then they breathe in the 10 microliters drumlet. And then we continue the dosing 10 microliters by 10 microliters. Because of the mouse nose has very small volume, so we cannot deliver large volume of mRNA vaccine. This is one limitation. Yeah. Oh, and then about your second question, uh, I think we haven't tested the other um, disease, uh, but basically we confirm the fluke mRNA, also the SARS-CoV-2 antigen encoding mRNA can be delivered by nasal route. So we believe if we test the other mRNA, if the encapsulation is good, and then we believe nasal delivery will be successful. Yeah. But actually, as you saw in the data, uh, delivery of mRNA does not guarantee effective vaccination. So, for example, IgA and IgG production is another story. So, actually, to confirm the effect, we really need to do actual experiment. Mm. Thank you. Mm.
네, 다음은 봉현수 학생 질문하시면 될 것 같습니다.
안녕하세요. 혹시 잘 들리시나요?
예, 예.
아, 제가 비전공자라서 그, 약간 제가 이해했는지 중심으로 여쭤보려고 하는데요. 그, 그 기강으로 mRNA 백신을 넣을 때 그게 이제 원래 백신이 단백질에 의해서 분해되지 않게 하기 위해서 PEG라고 하는 코팅으로 이제 그 분해되는 막는데 이게 mRNA 백신을 되는 게 효과적이지가 않아서 예, 박사님께서 ILNN이라는 걸 개발하신 걸로 이게 이해를 했습니다. 근데 이게 그럼 그 ILNN이라는 그 기술이 그 PEG라고 하는 다른 그런 코팅과 유사한 다른 코팅 기술을 이제 개발해서 적용하셨다는 건지가 좀 궁금합니다.
Yes, yes. So he asked the use of a PEG coating on ILLN2 system. So actually, ILLN2 also has a PEG, but it has a lesser amount. For example, I showed you we need at least a 5 weight percent of PEG to get effective coating. But in that case, mRNA transfection is very, very poor. So we try to reduce the PG coating to 2%. So you can see DSP PG is a pegylated lipid was added at 2%. So this 2%, we found because PG is still needed to stabilize the nanoparticle, as you mentioned. We try to add a minimum amount of PG at 2%. 네. Thank you.
박사님,
감사합니다.
저희가 지금 6명의 질문이 더 남았는데 괜찮으신가요?
저는 괜찮습니다.
괜찮습니다.
네.
괜찮아요. 제가 한 가지만 지금 손들기 한 학생들 이외에 이제 손들기 하지 말아주시고, 제가 학생들한테 한 가지만 부탁드리면, 지금 질문이 너무 많으니까 비슷한 앞에서 답변을 이미 해주신 유사 질문을 하실 분들은 사실은 답변을 이미 받았기 때문에 손들기 버튼을 좀 삼가해 주시고, we have so many questions, so please refrain from asking similar question, please. 네.
예.
그러면 다음 진행하도록 하겠습니다. 지금 보이는 순서대로 호명하겠습니다. 변희진 학생 마이크 켜시고 질문하시면 될 것 같습니다.
일단 좋은 강의 감사합니다. 저는 현재 엑소존 관련해서 공부하고 있어서 더 흥미롭게 들을 수 있었습니다. 먼저 일단 캐다이오닉 레피드를 사용하실 경우에 제타 포텐셜이 낮아가지고 어그리게이션이 높아질 수 있을 것 같다고 생각했는데요. 혹시 그런 현상은 없으셨는지 궁금하고, 또 PEG 코팅을 하면 LNP의 점액층을 통과하는 전략에 대해 말씀해 주신 걸로 이해했는데요. 반면에 엑소솜을 포함해서 많은 생체 유래 입자들이 다른 표면에 있는 특정 인자들에 부착해서 머무르는 시간을 길게 하는 특성을 사용한다고 알고 있는데, 혹시 비강 백신 개발에 있어서 점액을 빠르게 통과하는 전략과 점액에 머무르면서 약물을 서서히 방출하는 전략 중에 어떤 게 더 면역 반응 유도에 유리하다고 생각하시는지 궁금합니다.
Yes, yes. So her first question was about the zeta potential of a nanoparticle. So as you can see here, so with increasing the DOTMA to ALC 315 lipid ratio, we found the zeta potential gradually increased. This is because of the addition of more and more the cationic lipid. And then in the case, the pKa also increase. Then they try the surface charge become more positive. Yeah. About your second question, you mentioned about pegylation of exosome to enhance mucous permeability. Am I correct? Yes. Yes, yes. Oh, and then you also mentioned the comparison between mucoadhesive versus mucopenetrating system. So actually, you in the field, there were long time this debate, um, mucoadhesive versus mucopenetration. So and then only recently, I think about 3 years ago, papers are coming out, and then they revealed it. In the case of a small molecule drug, mucoadhesive property is helpful to deliver small molecule drug to the um, across the airway mucosa. But when we deliver large molecule drug like mRNA, in the case mucoadhesive is not so good because after mucoadhesion, the entrapped particle can be easily removed by the mucociliary clearance procedure. So if they stay, if the nanoparticle strongly bound, strongly bind to the mucin protein. In that case, they um, cannot stay and cannot have a high chance to penetrate and deliver mRNA. So they are easily removed by mucociliary clearance. So it depends on which drug you want to deliver. If you want to deliver small molecule drug, mucoadhesion system, okay. But if you want to deliver mRNA, mucopenetrating system is better. Thank you, thank you.
감사합니다.
And 에스카살람 샤피라 리즈키. Yes, your turn. Please turn on your microphone and ask. 샤피라 리즈키.
Can you hear me, Professor?
Yes, yes.
Yeah, so thank you for great presentations. I would like to ask about, so actually I have a pretty similar experiment, but I'm using DNA. But in I have like a compound tested in vitro, it can prove that it degrade my target protein. However, when I try in vivo, and it's quite hard to cell penetrating, so I add like more with the arginine so it can more cell penetrating, but I have still trouble in penetrating, so I'm curious about the how to do the PAG, like how to add the PAG with the RNA or how the mechanism. Thank you.
Oh, okay. Yeah. You asked the mechanism of integrating PEG lipid. So okay. In the case of I'm not so sure the composition of your nanoparticle,
Yeah.
but if your nanoparticle is based on the lipid assembly, I think you can also use a similar approach. So in our case, we dissolve all the lipid component in organic solvent and then make emulsion and then make a very small emulsion by emulsification and then evaporate solvent. So in that case, the hydrophobic interaction between lipid component it becomes stronger and stronger during the evaporation. And then in this process, PEG lipid is incorporated to the surface of a nanoparticle by in by through the interaction with the other lipid on the surface. Yeah. So probably in your case, you may have to dissolve your PEG lipid and other lipid in some organic solvent, for example, ethanol or methanol or some other solvent, and then need to evaporate together. In that case, you can induce a self-assembly of a lipid to make a nanoparticle.
But I have still to use the, like, for example, transfection using lipofectamine or something like that, or I don't need to do that.
Uh, does your system contains any uh, ionizable or cationic lipid?
Yes, it has
Uh,
like more lipid.
okay, okay. In that case,
I don't
the
think so.
lipid
Maybe.
if you add such lipid in your system, they usually
All
promote
right.
endosome escape and then
Thank you, Professor.
increase the transfection. So I think you don't need the other lipid. Mm. But maybe you need to adjust the ratio of ionizable lipid or cation lipid with the whole total lipid. Yeah, together. Yes. Hope you can get success. Thank you.
And next, Wanjara Baraka Robbins. Please turn on your microphone and ask.
Okay. I would like to ask a question. With reference to observations from the mice model, if the drug were to be used in humans, can you comment on how a drug delivery device would affect the nanoparticles' integrity and effectiveness? Let's say if we were to choose to deliver a drug by either an inhaler or a nasal spray or a nebulizer in humans, how can it affect the integrity and the effectiveness of a drug?
Yeah, good question. So actually, there are some papers about that. Actually, my system is different from inhalation. My system is a more nasal spray. So in the case of nasal spray, people already found the shear rate is very important. If the nasal spray device give a very strong shear force, mRNA LNP can be broke down. It's already published. So I think the most important thing is you need to make a stable formulation, and also you need to control the flow rate of the nasal spray. It should be mild in the case. Yeah. Mm,
Thank you.
thank you, thank you.
네, 이제 마지막 질문이고요. 정세명 학생 질문하시면 될 것 같습니다.
네, 교수님 좋은 강의 감사합니다. 들리실까요?
예, 예.
아, 네. 제가 발표하신 내용 중에 트랜스웰에서 뮤신 유사한 걸로 이제 투과가 실제 되는지에 대해서 실험하신 것도 되게 흥미롭게 봤는데요. 제가 뮤커스 가지고 실험을 하고 있어가지고 이러한 약간 너절 딜리버리 연구에서 실제 사람의 비강 분비물 가지고 연구하는 경우도 있는지 궁금합니다.
Ah, yes. So actually, her question is about the use of a real human mucosal sample for this kind to check the mucous permeability. Yes, some people in the US can do. For example, actually you can see the multi-particle tracking. Oh yeah, so you can see this multi-particle tracking technique. So they collected a real human patient sputum and then mixed with the nanoparticle and then measure the diffusion rate using the MPT technique. Yeah, but actually to do this kind of study, usually lab need to sign uh kind of the agreement. What is the IRB agreement? I usually in Singapore it's quite difficult to get IRB. I'm not so sure about Korea case. But if the lab can successfully have an IRB agreement with and then can recruit a patient, you can use a real human uh, you can use a real human sample. In my case, I use a pig sample. So actually from the sigma aldrich, we can easily buy the pig mucosa and then you can just dissolve pig mucosa in the cell line and then after we add the pig mucosa into the inner chamber of the transfer, and then after some incubation, usually 30 minutes, they naturally form a layer of mucin. Yeah. So and then the methodology is published in many papers. Maybe you can adopt such you can such methodology and then probably you can do in vitro test using the pig mucosa first. And then if you get promising result, and then maybe you can consider to move real human sample. Mm.
네, 그럼 교수님, 뮤신 유사한 것으로 실험하는 것과 실제 비강 샘플로 실험하는 거에 대해서 실제 비강 샘플을 이용해서 실험을 한다면, 임상적으로 좀 더 유의미한 결과라고 생각하실까요?
Uh, yes. So her question is about if we use a real human sample, is it considered more meaningful in the clinical value? I think yeah, I agree. If you use a real human sample, of course, it has a um higher value. Maybe you can how can I say the it's real human sample. We don't need to use animal sample to mimic, so it's more realistic. But I think in vitro test has some limitation. So maybe even if we use a human sputum, probably the system is not perfectly same with in vivo situation because there are a lot of environmental factor in the in vivo. For example, structure of a tissue, nasal tissue, structure of a tissue, and then the live cell, the live cell population and then how the epithelium move. Yeah, maybe many factors are there. So probably need to do real animal test if you really want to prove the value of your study.
감사합니다.
Thank you, thank you.
근데
감사합니다.
제 마지막인 줄 알았는데 한 학생이 한 명이 더 있어서 이 친구 아까부터 손 들었던 친구를
예,
한
예.
개만 더 받도록 하겠습니다. 임마누엘 아베베, yes, please turn on your microphone and ask.
Thank you. Thank you for the nice presentation. I just have one question. So my main point is that while developing a vaccine or any method in the vaccine industry, the ideal principle is to be safe. So when we see the safety profile of the safety profile, especially comparing the intramuscular route and that of the intranasal, how do you see that? Plus the thermostability, especially the mRNA vaccines are known to be not stable in the surrounding. They need a deep lower temperature to be stable. So does it make any difference in the route of administering that mRNA vaccine? That are my questions.
Mm. Okay, okay. So you mentioned about safety profile of a mRNA vaccine in the case of intramuscular injection and also nasal delivery, right? So I think okay, in my case in my lab we also test the intramuscular injection of a mRNA vaccine commonly. Usually in mice we don't see much side effect. Uh, it's also different from human case. For example, usually in human case, after intramuscular injection, fever usually come immediately and then last 1 to 2 days, and then also inflammation also happen in the human case, right? So, but mouse case it's different from human. So I think it's still animal models for vaccination study still limited. I think it's not fully recapitulate the real situation. Um, but at least when we tested in when we tested the intramuscular injection in mice, there is no specific side effect or body weight loss. Yeah, but only when we tried the nasal delivery, we found the serious inflammation in the lung, also systemic inflammation and body weight loss. Mm. And then, what was your second question? Second question was, uh,
The thermostability of the drug.
yes, yes. So actually, as you mentioned, traditional mRNA LNP has some poor stability. That's why when we ship the mRNA overseas, we need a minus 70 degree, very, very cold temperature, right? But actually, our system doesn't require such cold storage condition because our system is a ready to mix system. For example, let me see. Our system is we produce liquid core nanoparticle first, and then this nanoparticle is highly stable at 4 degree, and then uh, just before administration, we mix this system with the mRNA. So in this case, we don't need to worry about the stability of mRNA case. Mm. I think this is one advantage, yeah,
Yeah, I
of
think
our
that's
system.
one of the advantage. Okay, okay.
Mm.
Thank
Thank
you.
you, thank you.
Thank you. Thank you.
네, 백이현 박사님, 너무 제가 죄송하네요. 이렇게 사실 저희 지금 수업 거의 후반부 가고 있는데 이렇게 질문이 많았던 적이 사실 저희 역대급이었던 것 같아요. 처음인 것 같고.
그런가요?
네, 박사님께서 너무 잘 쉽게 설명을 해주셔가지고 사실 관련 있는 전공자나 비전공자 분들이 다들 이렇게 좀 관심 있게 들었던 것 같습니다. 저도 사실
아,
백신 연구를 안 하는데 굉장히 잘 재밌게 들었습니다.
예,
어,
감사합니다.
네, 시간이 너무 많이 흘러서 오늘 수업은 이걸로 마쳐야 될 것 같고요. 긴 시간 동안 질문과 재미난 수업까지 너무 많이 고생해주신 백이원 박사님 너무 감사드리고요. 김우석 교수님 그래도 아쉬우니까 마지막 코멘트 하시겠어요?
아니요, 아니요. 저 오늘 학생들 질문 많이 해주셔가지고 제가 궁금한
그쵸.
건 다 해결된 것 같아요.
네, 저도 사실
네,
질문할 거가 학생들 질문에 답변이 있었어가지고 해결했고요. 백예원 박사님 앞으로도 네, 계속 좋은 연구 잘 하시고 건강하시길 기원드리겠습니다.
아, 감사합니다. 아,
네. 네, 그러면 오늘 이걸로 수업 마치도록 하겠습니다.
네, 고생 많으셨습니다,
수고하셨습니다.
박사님.
네,
네.
수고하셨습니다.